Hello, everyone, and welcome to the RNA Collaborative seminar series. Today, we have two speakers from the Alberta RNA Research and Training Institute. We have Dr. Zovoilis, who is an Associate Professor of Bioinformatics and Canada Research Chair in RNA Bioinformatics and Genomics at the University of Lethbridge, and he'll be giving our first talk today. Ethan, whenever you're ready.
Thank you very much. Can you hear me and can you see my slides?
Yes, looks great. We can hear you.
Great. So, first of all, I would like to thank the RNA Collaborative Seminar Initiative for the opportunity to present part of the research we're doing here at the ARRTI at the University of Lethbridge. I will start with some disclosures. So my lab is part of three institutes: an RNA institute, a Genomics Center, and a Neuroscience Institute. And this reflects the collaboration between these three institutes and the research that is done in my lab, which focuses on non-coding RNAs and RNA modifications, and particularly on SINE non-coding RNAs and their impact on regulation of gene expression. And based on next generation sequencing and bioinformatics approaches, we try to find how SINE RNAs are associated with aging-associated diseases in brain. So the problem of dementia is a problem that is associated with an aging population, especially in the Western countries and in China. And here I have some statistics from Canada, but this applies more or less to most countries in these areas, where we expect during the next few years really a tsunami of dementia cases, because dementia and subtypes of dementia like Alzheimer's disease are associated with aging, and practically aging is the only real factor that has been strongly connected with predisposition of developing these diseases. The problem is, however, that apparently not all of us are going to develop dementia or Alzheimer's disease. So the factors that are really are responsible for the development and the pathogenesis of these diseases are complex and unfortunately still unclear, despite large projects that try to identify loci in the genome predisposing to Alzheimer's disease. We have identified associations, but still we are not clear which is the main pathogenetic mechanism that underlies late onset Alzheimer's disease. And many of you may have heard about amyloid beta and tau proteins. These are protein conformation. Alzheimer's disease is a protein conformation disease because these proteins have been found to be in aggregates and generating cases of alpha-beta amyloid plaques. So this protein deregulation is one of the fundamentals of Alzheimer's disease. But the problem is that we know now that there are people that may have, for example, this amyloid plaques, who, however, are not going to develop Alzheimer's disease. So there are still many open questions regarding what's the real molecular pathogenesis and the real mechanisms that underlie this debilitating disease. And of course, it's no surprise that still we lack much of this information that is connected with AD at the genomic level, since most of the research the last decades has actually focused on the less than 2-3% of the genome that encodes either for proteins or for known structural RNAs. And we know that for many years, and especially for this audience, this debate is very well known, that has been a lot of discussion about the rest of the genome and whether it's part of a junk DNA. And again, there was no surprise that this debate has taken so long, since until 2005, the whole amount of genetic information that we have been able to deposit in GenBank was not more than 50 gigabases, which is in a sense what a PromethION chip today can produce in 48 hours, and a NovaSeq Illumina sequencer practically in a matter of few hours. So these new high throughput technologies have revealed through a number of consortium, and here I just have the example of the ENCODE, that the vast majority of this non-coding genome is transcribed. And for this audience, apparently we don't need more details how this has resulted in the explosion of the non-coding RNAs field. But one part of this non-coding genome that has received maybe a little less attention, although it constitutes a large part of the non-coding genome, is the part of the RNAs that are produced by repetitive elements. And initially, RNAs that are produced by elements such as SINEs, the short interspersed nuclear elements, apologize, have been described as kind of genetic parasites that proliferate within the genome, and the RNA that's produced is kind of an evolutionary garbage or remnant of evolution. So for a long time, RNAs that are produced by these repetitive elements have not received real attention. The elements themselves, for example, the ALU elements, have received attention because they kind of may encode complexity in the genome and generating new splice junctions. But the RNAs themselves have more been regarded as kind of leakage of the transcription machinery that kind of transcribed these RNAs, but was not of really any functional importance. And here is the context I'm going to focus today my presentation, and this has to do with cellular response to stress. And cellular response to stress constitutes a fundamental mechanism that is deregulated during aging, and particularly in case of brain. We know, for example, that an aging brain or a brain that develops Alzheimer's disease shows many irregularities in pathways that have to do with response to stimuli, and this is then translated into cognitive impairment and synaptic malfunction. But coming to something more basic before we get to the neural tissues, so coming to some kind of like fundamental types of stress, like for example, response to heat shock or response to hypoxia, we know since many decades that when this stress stimulus is applied to cells, the cell try to survive this stress stimulus, for example, a heat shock, and in order to survive the stress, they suppress transcription of a big number of genes, for example, genes that are housekeeping and they're maybe not be needed for some time, and they do this in an attempt to reserve and preserve energy and help the cell to overcome that stress. And what has been known for some decades, but was clarified actually recently by the Kugel and Goodrich labs, is that this transcriptional repression that takes place during response to some types of stress like heat shock is actually connected with RNAs that are transcribed by the B2, the SINE B2 repetitive elements. And particularly what they have shown is that the B2 RNA that's transcribed by these repeats can bind RNA polymerase II, and through this binding, suppress transcription of these cells. And this, I would say, in my opinion, was quite revolutionary because it was the first time that really for these SINE RNAs, a clear mechanism and a clear key role in regulation of gene expression was revealed. However, this work did leave a number of questions open, and particularly the questions that were left open have to do with immediate early genes, or more widely stress response genes, that we know that during that time of response to stress are actually activated. So the question was, how do these genes kind of escape this suppressive effect from the B2 RNA? And while I was at Ginny Lee's lab in MGH in Boston, what we have found is that actually B2 RNA does have an innate self-cleavage activity, which is accelerated in response to interaction with a protein called EZH2. So what we found is that actually for these stress response genes, B2 RNA actually already in the pre-stress condition binds RNA pol 2 and keeps them poised for activation, but not active. So this is a kind of a quick switch where you have the B2 RNA sitting on these stress response genes, and upon application of the stress stimulus, the B2 RNA, after interaction with EZH2, is processed, self-processed into fragments, which releases RNA polymerase and enables transcription of these stress response genes. So this is a kind of way to generate a kind of switch where upon the stimulus, the B2 RNA is self-cleaved, releases pol 2, and enables activation of these genes. And of course, this identification of processing by B2 RNA opens new possibilities. And the main question that on which I have worked since I left in this lab and I started my own group here in Canada is whether this SINE RNA processing, this mode of activation, could be actually connected with a human disease. And response to stress would consist the best biological context to test that, because as I mentioned before, B2 RNA actually is connected with activation of stress response genes. And in case of brain, and particularly a part of the brain that's called hippocampus, which is a primary target of Alzheimer's disease, we already know that many of the abnormalities that are observed during Alzheimer's disease have to do with abnormalities in the response of the hippocampus to external stimuli that they receive. And to try here, I just represent multiple levels between the environment and the genome, where we have signaling that in case of brain is reaching the neurons in hippocampus, and then the neurons respond to this signaling by changing gene expression in order to adjust to the new environment. And this is from a previous work where I have participated, where we actually know that in hippocampus, this response to different stimuli is connected with activation of gene expression. And you can see here on the top, we have two categories of mice which have been stimulated and for which we got RNA from the hippocampus, and every row represents a different mouse in two different categories, stimulated and unstimulated, and every column corresponds to expression of genes. So there is a significant number of genes that are activated in response to a stimulus. And this, it's not the only work that have shown that practically in the hippocampus, response to a stimulus elicits a cell response that includes changes in gene expression. But of course, this is only one part of the story, because when we went through and found which genes are activated in hippocampus, we found that in addition to many genes that are kind of pro-survival, we also have activation of what we call breaks of cell survival, or actually what is called kind of the guardian of the genome, like p53. So it's not enough to elicit this activity. It's also important to be able to control this, because if it goes uncontrolled, it may cause downstream, as we will see later, a number of problems. So it's exactly this circle that in case of aging and AD is disrupted. And what you can see here is previous work where we have shown that in healthy hippocampus, indeed, after 1 hour of application of stimulus, we have a first wave of these learning genes, which are most of them actually stress response genes. And at the same time, this is followed by, actually after a few hours, it's followed by a second wave of microRNAs and, as I showed before, p53 that generate loops to self-control this wave and result at the end, in the end of this wave, and return to pre-stimulus situation. So getting back, I mean, having these temporary waves seems to be critical for homeostasis in hippocampus. And this is exactly what goes wrong in case of amyloid pathology. Because when we tested the levels of this microRNA in p53, actually we saw that these levels were constantly up, which actually implied that there might be a problem in this initial set of stress response genes in this condition. And because we have been working with B2 RNA, and B2 RNA is connected with stress response genes, we actually check whether these genes actually are bound by B2 RNA in a way that is non-random and enriched compared to other genes. And what we did find is actually that many of these learning-associated genes were actually genes that are bound and regulated by B2 RNA. And it was no surprise. For example, here we can see the learning genes versus all genes and their enrichment in B2 RNA binding sites. And when we actually check the B2 RNA regulated SRGs regarding gene ontology, the functions that are returned are very highly associated with activities that have to do with learning, like postsynaptic density, receptor localization to synapse, learning, synaptic transmission, and so on. So B2 RNA was a good candidate to be deregulated in our context. And again, as I said, we have the normal hippocampus. We do question here whether SINE RNAs are actually connected with this deregulation we see in AD, and particularly with the main pathologic finding of AD, which is amyloid beta pathology and amyloid beta plaques. To start answering this question, we first have to show that indeed the transcriptional deregulation we have been assuming because of the constantly high levels of genes that regulate learning-associated genes was really true. In other words, we have the assumption here that stress response genes regulated by B2 could be upregulated, which would have caused increase in levels of these genes, but was this really the case? So what we did is that we compared mice that are healthy, the wild-type mice, and then mice which have a humanized mutation for the APP genes, which causes amyloid plaques after the 6-month-old of age. And you can see here the deposition of plaques between the wild-type mice for these 3 time points and the APP mice for the same time points. And you can see that at around 6-month-old, the APP mice have clear deposition of these plaques, and it's exactly this time point of 6 months where these mice develop cognitive impairment and learning difficulties. So we took RNA out of the hippocampus at these different time points, And we checked, first of all, the levels of p53, and most importantly, the levels of the B2 RNA regulated stress response genes. And what we found is that actually in the APP mice, especially at the 6-month-old, during the active neurodegeneration phase, we actually have a large increase in the expression of these B2 SRGs. And in a sense, this was not a surprise, okay? So you have cells that are subjected to cellular stress, to neurotoxicity, because of the high load of amyloid plaque. So the cells try to respond to this problem by activating some pro-survival genes. But apparently, having these pro-survival genes high for so long activates p53. And p53 is connected with increased cell death. And at the end, what we know at 12-month-old is that neurons do die. So we have the wave, but the wave is persistently in high levels, and this leads to a general deregulation. Now, the question was, what leads to this abnormal activation of pro-apoptotic pathways? And as I said, B2 SINE RNA was a good candidate to check. And this is a work that was done by 4 great students in my lab, Luke, Yubo, Chris, and Babita. And what they did is actually taking RNA from these 3 different time points and subjecting them to a type of RNA-seq we have developed, which is particular for SINE RNAs, because these are shorter RNAs. They're more than microRNAs, so we can not do only small RNA sequencing, but they're shorter than the standard RNA-seq. So we went somewhere in the middle for something that is above 30 and below 200 bases, so that we can study these B2 RNAs and their fragments. And what we did find is that, as we have shown in the Cell paper, the B2 RNA is also processed at the same locations in the hippocampus of mice, and particularly in APP mice, it is highly processed compared to the wild type. And here you can see the mapping of the B2 fragments compared to the full-length B2 RNA levels between these 2 conditions. So we didn't find any difference in the full-length levels. We did find, however, in the process. So B2 RNA is actually highly processed in APP mice, in the mice with amyloid beta. And here you can see the quantification of this processing rate, which follows pretty well the changes we saw for the stress response genes. So at the 6-month-old, we have high processing rate, and it was no surprise that the genes that are regulated by B2 RNA are also highly expressed, because the B2 RNA is processed, it cannot bind any more RNA pol 2, and these genes are hyperactivated. Why, however, do we have this increase in B2 RNA processing? In the Cell paper, we saw that EZH2 was one of the factors that was actually accelerating processing of B2. But the problem is that the mouse hippocampus, it's not really enriched in EZH2. So this might not have been a good candidate, because if you don't have something practically, it's not likely that will be the reason for the effect we observe. I will not come into details how we get into the HSF1, but we did find that another critical factor for stress response, the heat shock factor 1, is actually highly enriched in hippocampus. And when we checked chip sequencing data and their distribution with regard to the B2 RNA regulated stress response genes, we found that after stress, these locations are actually enriched in binding of HSF1, and in our mice also at exactly that time point of 6-month-old, HSF1 expression was actually higher in the APP mice. Of course, this was in vivo. We want to check them also in the cell culture model. So we took a hippocampal cell line, we targeted HSF1, and at the same time, we also transfected this with toxic beta amyloid peptides to see what's the effect of the beta amyloid peptides and how changing the levels of HSF1 could change the processing rate of B2 and the expression level of the B2 stress response genes. And indeed, applying the anti-HSF1 abrogated this increase in B2 RNA processing. It also abrogated the increased levels, of course, of HSF1 and the increase in the B2 stress response genes expression levels. And to make the long story short, what practically these results depicted is that when we compare the healthy hippocampus with the amyloid pathology hippocampus, we see a very similar story with the one we saw during HITSO. We saw how, however, we have the same key player, B2 RNA, we have different interaction partners, though in this case, HSF1. So normal genes during stimulus, B2 RNA is processed. They return, however, to the post-stress during the recovery phase, and B2 RNA again stops transcription of these genes. What happens in APP and in the active neurodegeneration phase is that actually we constantly have, due to high amyloid load, high HSF1 levels, and we never really get to this post-stress recovery phase. That is what we found in case of mouse. How about human? And this is a work where Yubo and Luke in my lab invested most of the last 2 years, and we did found that SINE RNAs of the ALU class, which again have been shown by Kugel and Goodrich labs, that they are also blocking RNA pol 2, that they have also similar characteristics, suppressing transcription after HITSO, that these ALU RNAs are also processed in the brains of human patients. And here you can see the structure of the ALU, the cutting points that we identified in the ALU elements. And particularly here, we have been able to show this in a large cohort of patients from the ROSMAP study. And you can see here with red, we depict the 5 ends of fragments, and you can see that around this position here, we have a cutting point of ALUs. And when we check the processing ratio in between different categories with different clinical manifestation, one of them is no cognitive impairment compared to mild cognitive impairment and Alzheimer's disease. We also see, as in case of mice, increase in processing of the SINE RNAs. The same applied when we checked different classification based on pathological characteristics, and particularly Bragg states. Again, the more serious and severe the clinical outcome and pathological outcome, the highest the ALU RNA processing ratio. And of course, this is associated very well with p53 levels, which is again indicative of cell death. But the p53 levels associated only with the processing ratio, not with the full-length RNA, implying that the problem actually is the increased processing rather than changes to the full-length levels. Then we compared the processing ratio, which is here on the top, and the full-length levels, which is in this right panel, with gene expression levels of genes that are upregulated and downregulated in Alzheimer's disease patients. And indeed, the processing ratio between the no-AD and the AD patients correlated pretty well with the expression of these upregulated genes, and the levels of the full length that suppressed genes correlated very well with the downregulated genes. So again, I don't have time to go more on that, but this implied that in case of Alzheimer's disease, we also have increased ALU RNA processing, and this associated pretty well with transcriptome deregulation. Of course, because these are human patients, we cannot do the in vivo experiments that we could do in mice, but that's indicative that SINE RNAs tend to show similar characteristics regarding response to neurotoxicity in both human and mouse hippocampus. And here is kind of an overview of what we have learned regarding gene expression and SINE RNA processing in brain. I have here the example of B2 RNA, but in a sense, what our data showed is that actually gene suppression and gene activation are the opposite sides of the same coin, where we have, on the one hand, housekeeping genes that during stress do get silenced because of the high levels of SINE RNAs, while at the same time, stress response genes do get activated because of increased processing of those SINE RNAs. And with this, I would like to thank people in my lab, and of course, first of all, the students that performed this work, as well as the other members of the lab that supported them. I would like to thank also the collaborators. We have here the Neuroscience Institute, the Calgary Brain Bank, and the ROSMAP Consortium, and of course, the funding agencies that have supported this work. And I'd like to thank you again, and I'll be happy to receive any questions.
Thank you so much. That was a great talk. If any of the attendees are interested in asking a question, you can turn on your mic, or you can let me know in the chat or in the Q&A. If you have a question. I have kind of just more of like a random technical lab question. How do you actually prepare RNA from brain tissue?
Yeah, so I mean, initially we extract the part of the brain that's called hippocampus. So there is a way to prepare this. And then we just follow standard triazole extraction. It is a little more vigorous at the beginning, the homogenization process. So we do have to homogenize well the tissue. But because hippocampus is a tissue that is not high in fibroblasts, it's very easy to get homogenized. And after that, we just follow the standard protocol of triazole with chloroform and isopropanol precipitation.
Okay, that makes sense. I have a question in our question and answer box. So Nick is asking, could you comment on the potential role of SINEs in other chronic irreversible events such as fibrosis?
Yeah, thank you very much for the question. I find that this could be a possibility. Unfortunately, until now, we have tested only the case of amyloid beta pathology in my lab. SINE RNAs, however, have been connected with macular degeneration. And that was actually one of the first works that connected the ALUs with human disease 10 years ago. So I wouldn't be surprised that other similar contexts where you have chronic stress, transcriptome deregulation may be connected with SINE RNAs. But we haven't tested another context yet.
Awesome. So we have Akira. Do you want to unmute yourself and you can ask your question?
Sorry, it was my mistake of hitting the wrong button. Sorry.
That's okay, no problem. We have a couple other questions. So Marina, she asks, I have two questions. One, do you know more about the mechanisms of B2 degradation in response to stress? And two, are similar mechanisms acting in other tissue types / developmental stages?
Yeah, so regarding the mechanism, that's a work that was part of the Cell paper and the PNAS paper then that was published by the Lee Lab, is that actually we have self-cleavage. So the B2 RNA will be destabilized even in the absence of EZH2 or in this case HSF1, but the interaction with the specific proteins accelerates this processing. We have tested this regarding the tissue question in NIH33 cells. It was tested in HeLa cells. Now we have tested this in various neural tissues, and in all of them we find proof of this processing. We haven't tested other tissues outside from brain, but I would be surprised if they were not also so a certain degree of processing.
Okay, we're going to go one more question with Nils. Niels, if you want to unmute yourself, you can ask your question.
Yeah, theoretically, it's already unmuted. I don't know for sure, but can you hear me?
Yep.
You're
Okay.
good.
Hi, yeah, this is Niels Porter. Very nice talk. I'm following up, I guess, on Maria's question, which is, so if you say the cleavage itself, cleavage, does that mean there is an RNA enzyme activity? Is there ribonuclease activity? What's the actual enzyme or catalyst of the reaction? And why is it then stimulated by proteins? And ultimately, can you rule out that it's not something that happens post hoc during the prep process, for example? Because, I mean, of course, you expose your RNA to all sorts of things once you lyse the cells.
Yeah, these are absolutely great questions and valid comments. And these are actually the questions from the reviewers that have kind of quote tortured us for a number of papers. I mean, the Cell paper and the recent papers where we have in eLife and EBO reports, all of them had these questions. So we have included controls where we found, for example, that under RNAs with the same preparation were not processed. B2 RNA in presence of other proteins was not processed. And B2 RNA was not processed when, for example, we also denaturated HSF1. And of course, we have the PNase paper from Jean's lab, where she actually showed that both B2 and SINE RNAs do have this self-cleavage activity. So this is independent from an RNase activity, independent from the EZH2 interaction, and just EZH2 accelerates this. Which is the mechanism for this acceleration? We don't know. I mean, it's reasonable to assume that because these are RNA binding proteins, the interaction of the B2 with these proteins may help B2 receive that conformation that is more favorable for leading to this cleavage. But we have been able to exclude the fact that we have RNases or degradation as the reasons for this cleavage. But B2 RNA can by itself get processed. And if we make, for example, mutations, site mutations in the B2 RNA sequence, this potential is abrogated. So we know that the region between 98 and 105 is important for that, but we don't know the exact mechanism, whether this follow a non-ribozyme activity or non-class of ribozymes activity. So this is still not clear.
Okay, thank you.
Awesome. Okay, we're going to have to move on to our second speaker. Thank you so much, Dr. Zovoilis. If you have any more questions for Ethan, please just keep typing them in the question and answer box, and Dr. Zovoilis can go through and answer them as Luke's talking. Next up, we have PhD student Luke Saville from the University of Lethbridge, Department of Chemistry and Biochemistry. So take it away, Luke.
Thanks. I'll just quickly share my screen and get started here. Yeah, so as Laura mentioned, I'm a PhD student in Ethan's lab. I feel like you probably saw all of my different haircuts over the years in that presentation. I've got a new one now, so that's fun. But yeah, I'm a PhD student at the University of Lethbridge. And so I'll be doing a talk here today on NerdSeq, a novel approach of nanopore direct RNA sequencing that expands representation of non-coding RNAs. This is kind of a continuation of some of our work previously, some of which Ethan just presented, and actually we'll be using describing some of the sequencing methods that we're doing on some of the samples that we were working with in those two studies, or just sorry, the mouse study there. So I'd just like to say thank you for having me today to give a talk, and thank you for everyone to show up, or for showing up, and yeah, hopefully like our presentation. So NerdSeq is a protocol or method we've been developing in the lab, and it's sort of kind of an iteration on the direct RNA-seq method for nanopore sequencing. And so why we really like nanopore sequencing and what's really interesting about it is that it allows for the sequencing of full-length, unmodified DNA and RNA strands. Of course, it also allows for sequencing of, say, for example, reverse transcribed RNA or amplified by PCR DNA. But really what we are quite interested in our lab is looking at native strand sequencing, so specifically native RNA. And we're also quite interested in what kind of RNA modifications occur in RNA itself. And some of those being N6A, 8I edits, that kind of thing. And so how nanopore sequencing kind of works is that it generally works by ligation chemistry. So you have a read, it's ligated to adapters. One of the adapters has a protein on the end, and then that protein adapter binds to the nanopore protein that's sort of in a membrane, in a sort of a buffer solution containing a lot of ions. And so as the strand is pulled through the pore, it kind of perturbs the ions and creates a current. And then you can kind of create something called a squiggle plot. And so how that kind of squiggle plot works for a whole strand, it can be quite noisy. So there is sort of a neural network processing that kind of goes through the squiggle plot and calls specific changes in the plot as different nucleotides. So you have an A, C, T, G, U, and has the potential also to base call any sort of minor changes to that squiggle to call it as perhaps a modified base, such as I said, N6A or 8I or that kind of thing. So yeah, but the interesting thing about that method is that the standard approach, being based on ligation chemistry, largely relies on capturing poly A strands. So typically just you get mRNAs and some polyadenylated non-coding RNAs. And as Athan mentioned in his talk previously, so in the, say, mammalian genome, about 74 to 90%, depending on the genome, is constituted by non-coding regions, and that's largely transcribed. And so, as for the topic of his talk, which was on SINE RNAs, for example, which are repeats throughout the genome, these are non-coding RNAs that in some cases are polyadenylated and sometimes not, but some of those would, in the sort of standard sequencing approach for nanopore direct RNA sequencing, would be precluded from the reads. And other interesting non-coding RNAs would also be excluded. So this is just sort of what the standard direct RNA-seq approach looks like. So you have, in a pool of RNA, you would have some polyadenylated transcripts, so mRNAs, for example, and some non-coding RNAs that have a polyadenylated tail. And then you do a poly-T adapter ligation onto that polyadenylated tail. And so that would exclude some of the transcripts that you don't necessarily want, such as ribosomal RNAs, which constitute large portions of the transcriptome. But then you may actually get rid of some RNAs that are quite interesting, so such as SNO RNAs, such as tRNAs, or other things that you yourself may be interested in. And so after the poly-T ligation, they do a first strand synthesis. So this isn't the typical RNA-seq ARRTI step, where you would do a first strand synthesis and then you would add more adapters on certain ends and then do a second strand synthesis, and then you'd have full cDNA. In this case, you create an RNA-DNA hybrid, the DNA acting as the backbone to the RNA, allowing it to more easily run through the pore. And it also helps to stabilize the ends for more accurate sequencing. So as it goes through the pore, if it's wiggling too much, it could make base calling much more difficult. So if you have a backbone, it makes it a little bit more linear. It's a little easier to get a nice, more clear squiggle plot for base calling. And then finally, you ligate onto those end adapters, the sequencing adapter, and that's what kind of contains those proteins that I mentioned that will eventually bind to the nanopore itself, and then it'll help to pull the strand through the nanopore. And so I'm just going to quickly describe to you what we've sort of modified in our NerdSeq protocol. So it's non-coding enriched RNA direct sequencing. Always got to fit an acronym in there. And yeah, so generally when you have your pool of RNAs that you want to sequence, so like for example, here's our pool, and we have some short non-coding RNAs here and here, for example. So instead of where we would previously exclude them, we actually do a size selection, quite similar to the approach that Athan described in his talk for our mouse brain sequencing with Illumina. But instead of sequencing the short and long fractions separately, we simply polyadenylate the short fraction and then pool the long and short fractions together. So you have now essentially what was your pool before, with more so your short non-coding RNAs containing polyadenylated tails, and then you have ribosomal RNAs that are not necessarily being polyadenylated because they would remain in the long fraction, assuming your RNA quality is good enough. And then we do sort of a random priming after the poly-T adapter ligation onto those newly polyadenylated tails, and then of course the transcripts that already contain polyadenylated tails. And so the reason behind the random priming is primarily to sort of stabilize the read as it gets reverse transcribed, because what we were finding is that a lot of these interesting transcripts on the short end, so specifically things like SNO RNAs, which are extremely structured, weren't being represented in the reads very much. And then when we added random primers, they were better represented, which we thought was an interesting innovation. And then when we did reverse transcription, one of the important things that we also think that we found was doing a high temperature first strand synthesis instead of the normal sort of superscript style first strand synthesis on the RNA strand, seemed to also include those more complicated structures in the reads as well. So we used, instead of say superscript, for example, we used a LAMP enzyme called OmniAmp from Lucigen. And this does a reverse transcription at about 70 degrees Celsius. And it's also characterized to have strand displacement activity. So it's able to sort of get through some of those more complicated double-stranded regions. And then we do sort of the standard end adapter ligation, and then it can be sequenced through the port. And so some of the data that we've been getting out of the reads is that we generally have similar length to sort of the standard method, so standard in gray, and then NerdSeq in red. But we have general changes in the enrichment patterns. And then comparing non-coding to coding representation, we do see better non-coding representation in these reads compared to the standard protocol I mentioned. Additionally, we see better enrichment of sort of reads that appear more intronic. So that would include a lot of non-coding RNAs, for example. We do have some enrichment of line elements, or reads that map to line elements, for example. And then when we look at specific classes of RNAs, for example, just producing metaplots, we do see that although mRNAs, which would already be polyadenylated, are not as enriched in the NerdSeq protocol compared to standard, so NerdSeq in red, we do still get pretty good representation of mRNAs. And then I'm not showing, I don't have it here, but when we show a more relative sort of scaled graph, it does have a similar profile as well. So we're getting similar read lengths as well. And then when we look at non-coding RNAs with a polyadenylated tail, such as B2 RNAs, we're also getting that representation, although not necessarily as enriched. But then when we move on to the non-coding RNAs that don't have a polyadenylated tail, the enrichment is quite significant. So when we have SNO RNAs, for example, we see quite a large enrichment. Same with SN RNAs, same with SC RNAs, and then same with SRP RNAs. And then we also see enrichment of tRNAs. And then kind of an interesting thing that we saw when we mapped to ribosomal RNAs is that we don't seem to be getting full length. So just on the metagene plot here, we don't seem to be getting full length ribosomal RNAs. We seem to be getting these like short fragments. And then when we map them, it seems that we're actually enriching for what has been previously described as ribosomal RNA fragments. And these seem to be some interesting emerging short RNAs that kind of act in ways kind of like microRNAs, where they sort of modulate ribosomal RNA transcription, So that's something that we've been quite interested in starting to look into. And then we also see an enrichment of line 1 or line RNAs, so reads that out to line RNAs. But this doesn't really include line L2, more so just the line L1 element. So just the conclusion from this, we've from our method, we've developed a sequencing strategy that enriches short non-coding RNAs in direct RNA-seq libraries. Some of the examples of reads that or RNAs that we can enrich for with this are SINE RNAs, SNRNAs, scRNAs, SRP RNAs, ribosomal RNA fragments, tRNAs, and other non-coding RNAs while maintaining mRNAs and other natively polyadenylated transcripts. So future directions with this will be to explore other tissues. So this was working entirely in hippocampus. We'd like to expand to other regions of the brain and then other tissues as well and see if we're finding any fairly interesting non-coding RNAs that might not even be described elsewhere or that kind of thing. And then further, we'd like to evaluate RNA modifications in native RNA data. This is sort of an emerging challenge for Nanopore as some of the modifications might be kind of discrete. So not very apparent in that squiggle plot. So looking at ways to deconvolute that and try and compare in the short RNA reads as well is of further interest. So that's the end of my presentation. Just like to thank members of the lab, specifically those who also worked on this project, like Dr. Athanasios, a previous lab member, Dr. Levita Golan, my colleagues Liam Mitchell and Matthew Stewart Edwards and Travis Haight, and then our newly graduated and recently moved on to a different job, Dr. Yubo Chang, who did a lot of analysis for us as well. And then some of our great collaborators, Dr. Majid Mahajarani, who did a lot of the work with the mice for us, and then Dr. Jugender Mala, who did a lot of the extractions of the mice for us. So that's the end, and yeah, happy to take any questions.
Awesome, thanks so much, Luke. So my favorite RNA modification, pseudouridine, I don't think you mentioned it at all. I'm assuming it's one of the harder ones to probably detect because it's, yeah, so similar.
Yeah, well, I mean, we haven't done a lot of work with doing the RNA modification detection in some of these reads. This is kind of a newer project for me, so we're working a bit early on this one. And but pseudouridines, there has been some, some literature on doing pseudouridine modification detection in Nanopore reads, but it's a bit challenging to do all of them at all times at this point, but I think there's been quite a bit of work on that as well.
Nice. Okay, so we have a question from Alessandro. He says, "Hi Luke, nice talk and very nice technique. Are you able to detect pi RNAs too with NerdSeq?"
Yeah, I think so. I can't remember if we looked back at that. I'd have to look back at the data sets that we were working with and how we were annotating those to confirm. So I know it's not a very satisfying answer, but I believe so, yeah. Nice.
And then Clement says, how you size select your ncRNA? Is it gel size selection?
Yeah, sorry, I guess I should have mentioned that. So it's it's like a column kit. So I think it's called the Invitrogen Mirvana kit. So it just sort of, it basically you get different size fractions, and then you can do purifications with the same column, or sorry, a different column. So you have your short and long separated, and then purify them separately.
And then I think they had a follow-up question. It was, when they're selected, do you just poly-A them with poly-A polymerase?
Yeah. So just the E. coli poly-A polymerase, yeah.
Cool. Okay, Camilla says, great talk. What is the average percentage of non-coding being sequenced full length?
I think I understand your question, but I think we had a slide on this. And sorry, I went kind of quick just so I didn't go over time. But we do have sort of so our non-coding graph here is our read coverage is about 20% non-coding and then 80% mRNA compared to the standard, which has 5% non-coding and then about 95% coding.
So good.
I think so.
Did you ever look, Colette asked, did you ever look individually at intronic reads? I saw they were combined with intergenic reads in your plot.
Yeah, I think this was, I didn't do this graph, so I apologize if I don't give a super satisfying answer here, but essentially when we're developing annotations for this comparison, we were just going sort of through like the UCSC table browser and getting, basically looking for whether we were getting mapping in sort of those intron intergenic regions. And I don't believe it was easy to separate them out, but I didn't do that analysis. So I apologize for a not super satisfying answer.
And then we have Frank Slack. Do you want to ask your question?
Yeah, hi, Luke. Just following up on the piRNA question, I'm wondering about microRNAs and also isomers, the variants of microRNAs. Were you able to detect those?
Yeah, I think we looked at those, but it's possible that with the alignment software, so it's just like the Nanopore Minimap one, that I think that we weren't able to get very satisfactory mapping to those elements. I have to look back again though and confirm, similar with the piRNA, sorry. But yeah, I believe it wasn't very clear, no.
All right, thanks.
Yeah.
All right. Andrew asked, can you deploy adenosine analogs during the poly A addition step to differentiate between native poly A tails and those that were added during this process?
Yeah, I think you could. The one change that you would also have to do is change that adapter that Nanopore provides because it is a poly T. So say you were, or to sort of add a pool of say like a poly C or something like that, and do like a poly G tail or something. I don't see why not, or even doing modified basis like inosine. Yeah, I don't see why not, and I think that would be an interesting way to change that up. Yeah, for sure.
And then Logan asks, nice talk. I am wondering how much of an improvement random priming provides NerdSeq over the standard RTA priming.
Yeah, I don't have the figure here today because it was a little while ago when I was looking at that. But generally, when we were doing random priming versus no random priming, we just weren't getting the same enrichment. So we were getting much better enrichment when we random primed. And again, I think that maybe just because it's helping to sort of kind of help aid in the unwinding of those reads. Not very clear on what are the kinetics of that change, but it just seemed to improve enrichment generally.
And I think Logan also had another question that have you tried to determine if any of your mRNA reads are nascent RNA or mature RNA?
Yeah, we haven't looked at the distinction so much, though I can't imagine we would be getting a whole lot of nascent because we're not necessarily doing or we're not doing the polyadenylation on a longer fraction. If there's some really short nascent mRNAs, perhaps. We haven't checked that though.
I am not sure if I'm going to read this question right, but it's from Chin. What is the difference versus regular RNA-seq? Or if you want to ask your question.
I think I understand what is the difference versus regular total long RNA sequencing.
Okay.
Well, so first of all, I mean, generally, total long RNA sequencing, you would do that, say, on an Illumina platform, because with Nanopore, the preps are still requiring sort of that ligation chemistry. If you could find a way to sort of, ribonucleate and then just do total long RNA sequencing with those adapters in mind, perhaps. But yeah, I'm not sure if that answers your question. Oh, yeah, I think you edited it. Yeah.
And then how does the cost compare?
Yeah, I mean, so Nanopore is currently a little bit more expensive than Illumina just on the cost of the flow cells itself. So the cost is a little bit higher, but as that goes down, I think it will be improved. The direct RNA-seq itself compared to say like an Illumina sequencing is a bit challenging for a cost comparison because you can barcode with say like when you're doing the cDNA, for example. And you can do barcoding on cDNA for Nanopore, but not with the direct RNA-seq.
Okay, last question is from Colette, and she says, I'm surprised to see that there seems to be more short reads in the standard sequencing compared to NerdSeq. Plot A on this slide. This doesn't seem to support enrichment of short reads by NerdSeq. Can you comment on this?
Yeah, so I think there's a couple things to this. And we've been trying to figure this out a little bit too, is because we do see definitely enrichment in short RNAs. So yeah, that's a good question. And so we kind of see this sort of like crossover at about 250 where the read enrichments change. I think this might be a lot due to the mapping itself. So we're typically doing mini-map 2 for mapping, but we've been trying to optimize for short RNA reads. So I think that's because it tends to be a little bit more throughput with the standard read method. We are likely getting a little bit more just reads represented with that mapping strategy. When I do the sequencing itself on the machine, I do get like a GUI output. And typically we are seeing changes in the read distribution. I don't have that prepared for you today, so I apologize. But with standard, we tend to see a little bit longer as our N50. And then with the NerdSeq, it seems to be a little bit shorter on the N50. So I think there might be mostly due to the mapping itself.
Awesome. Thanks so much, everyone, for coming today. It was a great talk. Thank you so much again. And if you're interested in attending more of these RNA collaborative seminar series, the next one is April 20th with Cambridge RNA Club. So yeah, thank you so much again.
Thanks, everyone.
Have a great day.